Good evening, everyone. Thank you all for coming out tonight. Lovely to see so many of you here. My name is Beth, and I am new with the See and Learn team. First of all, tonight I want to thank the sponsor for our live stream. That's Carib Trans. They're making this lecture tonight and every night available through our Facebook live stream. So we are appreciative of that support. I should also thank El Momo Cottages, who are sponsoring tonight's presenter. So that's a huge benefit and makes See and Learn possible. We really appreciate their support. And also the Prince Bernhard Culture Funds and public entities SEBA, who support all of See and Learn throughout the month. So they make all of the activities that we do, the field projects, the activities with the schools and the senior center, and all of these evening lectures a possibility. And we actually have, in addition to those major sponsors, over 50 community sponsors that make See and Learn possible. So we do want to acknowledge all of their support as well. Now, for the next couple of days, just a couple of things that you guys should know about upcoming. So tomorrow night we have a night dive with Paul Sikkel, who's doing our presentation tonight. Come out and do a cool night dive with us where we're going to be collecting and observing some of the marine parasites that he's going to be talking about. Then on Wednesday, there's a snorkel opportunity in the evening or in the afternoon at 1 p.m. You can go out and snorkel with Dr. Ian Hewson, who will then be doing the presentation Wednesday night at Ecology on the diadema die-off, which has been happening all around the region and globally, and it's really important. It's some super interesting research that also pairs really well with Paul's research. So do come by, visit us at the See and Learn tent and learn more. If you participate in any of our field activities, you get a really cool See and Learn shirt that you've seen scattered throughout the crowd and people wearing them around the island. So just another reason to come take part. You can rep the brand, and everything is made with recycled plastic. You may have also noticed behind me and over in the corner and in lots of people's hands, let's see, raise a glass. Are you drinking a Saba Cloud Top right now? Yes, excellent. So feel on top of the world with Saba Cloud Top that does support See and Learn. I should mention before we move on to tonight's speaker that we also have amazing raffle prizes. So please do buy some raffle tickets if you haven't. We've got some really cool things available as part of the raffle, and they are only $5 per raffle ticket. So stop by and see us. Buy one, buy five, buy an arm's length of raffle tickets. So for tonight, we do have Paul Sikkel here, who's speaking on exposing the hidden majority, the ecology of host-parasite interactions in coral reef ecosystems. So Paul was actually last here about 10 years ago as one of our See and Learn experts. And this year we decided to bring him back as part of our 20th anniversary excitement. Paul is a research professor at the University of Miami's Rosenstiel School of Marine, Atmospheric, and Earth Sciences in the Department of Marine Biology and Ecology. His current research focuses on the ecology of host-parasite interactions in coral reef systems. Paul's work has appeared in a bunch of different media outlets, including National Geographic, both film and print, the BBC, including Blue Planet 2, which is very cool, National Public Radio, and Channel 1 News, which is a national news program for high school students, so he's reaching audiences of all ages. Unlike the topic, parasites, Paul's presentation tonight will definitely not suck, so let's find out together by welcoming back our tie-dye loving Paul Sikkel.
All right. Thanks, Beth. Thanks so much. Thanks to See and Learn for having us back. I'm really shocked that they would have us back, but we'll take what I can get. Really appreciate it. And also to El Momo for both putting us up and putting up with us. All right. So, if I were to take you on a little diving adventure on any coral reef in the world and ask you to give me your perspective on biodiversity, you would probably say, oh, there's a lot of cool fish there, or lots of corals, or maybe you're into macro, and so you'd point out all the, the invertebrates that you can photograph. So you'd focus on things that you can see. That's natural. We're, terrestrial primates. We're lucky to be able to go underwater at all. But if you look at each one of the things in this picture, and I'll focus on the fishes for now, but it applies equally to everything else. What you see are lots of little things that are living on and inside those. And so these are parasites. So everything in that picture has multiple species of parasites that live on it, some of them unique to that species. And even the parasites, some of them have parasites. So you can get a sense of how quickly the biodiversity of those organisms ramps up. It boggles the mind. And yet, because we're, at the scale of our vision, we don't see them unless we look specifically for them. Also, if I were to ask you for your perspective on trophic interactions in the ocean, those are the interactions that involve the transfer of energy. So what eats what? You'd say, well, yeah, big fish eat smaller fish, and those fish eat even smaller fish, and so on. But also, on the right, little things can eat big things. They just do it a little bit at a time. So it goes unnoticed. All right. So my focus of our research is on parasite-host interactions, and it didn't start off that way. I'll explain how we backed into that. But in writing grant proposals and getting our research published, we have to justify our existence. Like, why should anybody care about this stuff? So I'm going to tell you what kind of things that we point out as important aspects of these organisms and why they should support our research. So first of all, parasitism is the most popular biological profession. So all organisms have to obtain energy somehow. And the most common way of obtaining energy in the wild turns out to be parasitism, more than any other way of obtaining energy. It's globally, roughly half of all organisms make their living via parasitism. But in high diversity places like coral reefs, it's as much as 80%. So, mind-boggling. All right, so if you do a Google search, you type in food web. You probably remember this from elementary or high school food webs, and you have these complex diagrams with all these arrows of what's eating what. So you try and do that search, you'll see hundreds of these diagrams for all kinds of systems, open ocean, forests, deserts, mountains, whatever, but you won't see parasites in any of them. And yet everything in that picture that you see is from parasites. So when you start adding, when you start adding the parasites into that picture, it completely changes the picture. All right, as you know, parasites can cause or facilitate diseases, and therefore they can impact population dynamics of their hosts. That's important. They can influence behavior and how organisms utilize habitat. They can mediate other kinds of symbiotic interactions like cleaning symbiosis. They can mediate species invasions. They can influence whether or not an introduced species can become a successful invader. They can be used as indicators of habitat quality and the effects of humans on the environment. And they can be used to identify the source populations of hosts. So you can, by taking the parasites off of a host, figure out where it came from. So all the things in white are things that we do, our lab does. We haven't gotten into the yellow one yet, but everything else we do. All right, so how does this all began? So the most frequent question asked after people ask me, what do you do? They say, well, why do you do that? And how did you get into that? And so the story is this: I was originally trained as a behavioral ecologist. My focus was on reproductive behavior in fish, particularly damselfish. And in the Caribbean and elsewhere in the tropics, the damselfish spawn at dawn. So like at first light, as soon as there's a little bit of light, they start. And by half an hour past sunrise, it's all done. You've missed it. And so I spent months every day watching these fish. In this case, it's a yellow-tailed damselfish spawning at dawn. All right, how about here? Spawning at dawn. And so the problem they face is that the female has to go to the male's nest to spawn. She then has to leave her territory to do that. She has her own territory that has resources in it like food. She has to leave home to go to his place to spawn, leaving her place exposed. And so there's a trade-off there between spawning and territory defense. And what I noticed is that the females were interrupting spawning to visit cleaning stations near the male's territory. It's like, why are you like adding time to your time away from your territory? Why don't you just do the cleaning later? You know, why don't you just do spawning and then later get cleaned? So whatever it was that was causing them to go to the cleaner fish must be pretty important. And so here's a female getting cleaned by a cleaner goby. And so this story was told through in a film called Ripple, which was produced by Jennifer Berglund. Those deadheads in the crowd will get the reference. So you can do a Google search of Vimeo Ripple, and you'll see that film. It's about a 12-minute film. All right, so these fish are getting cleaned at dawn. And so what are the cleaner fish eating from them? That's probably the key to figuring out why this is happening, is that whatever the cleaner fish are picking off of them is probably what's driving them to do this. So what do cleaner fish eat? Well, if you cut open a cleaner goby in the Caribbean or a cleaner wrasse in the Pacific, they're full of these things called nathid or nathid isopods. These are, and there's like popcorn. They're just full of these things, hundreds of them in their gut, depending on how big the cleaner is. So this is their primary prey item, these little isopods that they're picking off the fish. That red you see in there is blood from the fish that thing fed on. This is obviously not to scale. These things are like 1 to 2 millimeters long. Yeah. But so these cleaner fish are eating these little isopods. So, all right, so when are these isopods active? Get to that next. So if you look at cleaning activity on a coral reef, then these are for another kind of damselfish. You'll see on the left there's a peak at dawn. So if you follow a fish throughout the day and look at when it's interacting with a cleaner, dawn is the peak. And it's like the first hour of sunlight. If you're in the water after that, what you see is that big drop off down on the right. So from maybe like 6:30 until maybe 3 or 4 in the afternoon, it's pretty low. And then at dusk, it starts to pick up again, but nowhere near what it is at dawn. Like dawn is by far the peak. So dawn is like the peak cleaning time for these fish. All right, so this is the title of the presentation, and here are our social media handles, if you care to follow us. Let's first do a quick review on symbiosis. Since we're talking about parasitism, which is a type of symbiosis, let's do a review so that we know how parasitism compares to other types of symbiosis. So commensalism is where both parties, one party benefits and the other party, no big deal, whatever. So no harm. Mutualism is where both parties have a positive outcome. That is, the interaction benefits both parties. And parasitism is where one party benefits at the expense of the other. That is, in engaging in the interaction, one thing benefits and the other thing experiences a cost. Now, if you want to get technical, mostly about the last one, but the second one is actually to come in as well. All right, most parasites are what are called cryptofauna. These are tiny organisms that you don't see, they're hidden, but there are exceptions, right? So some parasites are actually big, like lampreys. All right, so let me talk a little bit about the life cycle of these nathid isopods. They're unusual even among parasites in that only the juvenile stages feed, and there are 3 juvenile stages, and each one feeds on a single host. It feeds on a single fish. It then digests that blood meal, metamorphoses to the second stage, feeds again, digests that blood meal, goes to the third stage, feeds again. After the third feeding, and there's some blood-fed ones, after the third feeding, it becomes an adult, either a male lower left or a female lower right. So only the juvenile stages feed, the adult stages don't feed at all, they just reproduce and they die. But the taxonomy is all based on the male specimens. So you have to use the taxonomy to identify the species, you have to rear them up to get males. Right, so just like ticks and mosquitoes, they spend most of their time not on the host. They attach the host just long enough to get a blood meal, then they fall off and they go back in the substrate. And so the substrate has a lot to do with where they can live, what places are good for them or not so good for them. So they spend about 90% of their life not on the host. They just hit and run, take their blood meal off, and hang out in the substrate. So aphids are just like ticks and mosquitoes. We call them the ticks or mosquitoes of the sea. Just like ticks and mosquitoes, they feed on blood of their host, in this case fishes, and they don't stay on the host very long, just long enough to feed, then they either fly away or drop off. All right, so when are these things active? Well, if you look at the left green arrow, that's dawn, and then the right green arrow is midnight, and so you see that there's basically two peaks. As the day goes on and it gets to dusk, you start to see an increase, and that peaks around midnight, and then a little bit of a decline, and then another spike at dawn. So you can imagine a fish that's going to sleep at night, right around dusk, it's getting hit with that midnight peak, and then when it wakes up in the morning, it's getting hit with a dawn peak. So it wakes up with like all these things on it from the night before. And so you can see why they want to go to a cleaner and get them removed, even if they should be spawning or guarding their territory. It seems pretty important. If you look elsewhere in the world, this is the Philippines, you see the same pattern. In Australia, you see the same pattern. So it seems to be a global pattern that these things are most active between dusk and dawn. All right, what's also cool about this is that there's a difference in the sizes of aphids that are strongly nocturnal and those that are active more around the dawn and dusk period. The bigger they are, the more strongly nocturnal they are. I wonder why that happened that way. That's weird. Anyway, so the bigger they are, the more strongly nocturnal they are. So you almost never catch a small one around midnight, and you almost never catch a really big one in dawn and dusk. And this happens around the world wherever we look. But what's cool about it is in the Caribbean, this happens within species. This is the same species that as it gets older and develops, it becomes more and more nocturnal. Whereas in places like Australia, you have some more diurnal species and some more nocturnal species. Grunts and snappers is that at nighttime, they split. They leave the reef. They go out in seagrass beds or sand flats and feed, but they're off. So overall, nocturnal fish are more susceptible, but nocturnal fish that stay on the reef at night, they're pretty resistant. Nocturnal fish that leave the reef at night are very susceptible. And that has consequences. So grunts and snappers, mostly grunts, are probably the most studied fish in the Caribbean. A lot of work has been done on their migrations. At dusk, they migrate out to the seagrass bed or the sand flats, and then at dawn, they come back to the exact same place. You can go there every day and watch a group of grunts make their migration out, come back in. This is also shown in Ripple if you want to have a look at that. Nice video of that. So the hypotheses for why that happens is one, they get more food in the seagrass beds, or two, they reduce their risk of predation. But from what we've shown about how susceptible they are to parasites, this is another possibility. The third one is that they're basically leaving the hotbeds of the parasites in a way that reduces their exposure. So this is an experiment we did. If you look at the two bars on the left, those are fish that we put out at night or dusk and picked up at dawn. On the left is reef, and on the right, seagrass bed where they want to be. The reef is where they don't want to be. And then on the right ones is a much shorter set, which we set at dusk and picked up around midnight. So in both cases, you see that you get a lot more parasites on them if you make them stay on the reef than if you let them go in the seagrass beds where they want to be. So a huge difference. But what's equally important is that while they reduce their exposure to the parasites, they don't eliminate it entirely. And this has consequences because as they're leaving the reef at dusk, they get some on them and they transport them out and they fall off in the seagrass bed where they can then infect other fish. Then when they come back in the morning, they come back just after the dawn peak. They dodge that peak and they hit it on the way down, but they still get something. And so by that, they can effectively transfer energy from the seagrass bed to the reef because the parasites feed on them and then drop off on the reef. So they're trafficking aphids to the seagrass bed, and they're transferring energy from seagrass to reef by aphids that feed on them once they get back. So this is part of Gina Hendricks. Gina Hendrick is my PhD student back there. This is part of her PhD work. So what you can see here is you have this whole network of grunts on different reefs, all meeting in the same seagrass bed, and then gnathids that have been transferred by one grunt may later be picked up by another and transferred to another reef. So you get all this mixing of these parasites through this grunt migration, which is pretty cool. All right, what's another twist of this story is that most of you who've been diving for any length of time at all have seen these big isopods on the face of that fish, right? So those are more, when we mentioned we work on parasitic isopods, most people think of we're working on those. We do some work on those. And so they're much bigger than the gnathids, and they stay on the fish for about a year. Turns out that fish that have those big parasites on them rarely make the migration. They tend to stay put, whereas the ones that don't have that parasite on them consistently go out. So the ones that have the parasite tend to stay back, this big Antilocra isopod. All right, let's talk about effects of gnathids on settlement stage coral reef fish. So coral reef fish, as you may know, spend their larval stage in the plankton. They're hatched from eggs that are laid on the bottom, or the eggs are broadcast in the water column. Either way, the larvae end up in the water column and taken away by the currents. And then at some point, roughly a month from when they're hatched in the water column, they come back to the reef and they settle out. And they uniformly settle at night. And that has the benefit of avoiding all the planktivores that are active in the daytime. They can easily pick them off as they're settling on the reef. But we now know that there's other things that are very active at night that could actually do them in. And so these are gnathids that are attached to settlement stage fish, and depending on the size of the fish and the size of the gnathed, it can take as few as one gnathed to kill a settlement stage fish. So they can have direct impacts through mortality of these little fish. And even if they don't kill it, they can have impacts. So this is a heat map of two little damselfish that were placed in an arena. And damselfish don't like each other, they fight. And so we put a little chamber in the middle of that bolt. That's like the shelter hole that they both want. And then some were infected with a single gnathed, and others were not infected. And so these are ones that survived the infection or the bite by the gnathed. But even if they survived, if you look at the heat map of the uninfected one, it's basically bogarting that middle section. It owns that. Whereas the one that's infected is way out here on the edge. Like, it can't get close to that sweet spot. So even if it doesn't die, it loses out in competition for shelter spaces, which is very important in preventing predation, because those little fish need to hide from big fish that eat them. All right, so as I mentioned earlier, just like ticks and mosquitoes transmit bloodborne parasites, there's good evidence that gnathids do as well, particularly apicomplexan parasites, protozoans that are related to malaria, right? So some gnathed species, not all of them, have been shown to transmit these apicomplexan blood parasites. All right, so we've shown how these parasites can impact the host. There's a variety of ways they can have negative impacts on the host. But if there's only a few of them out there, it's not a big deal. Really, it's just a nuisance, no big deal. So really, the degree to which they impact their host depends on how many of them there are. And so what determines that? Right? That's an important question. So we know that cleaner fish eat gnathids. That's how we got into this in the first place, right? Is by looking at cleanering interactions at dawn on damselfish. So they eat those things, but they only eat them during the daytime. They're not active at night. So what about the gnathids that are active at night, which is actually the majority? So nocturnal planktivores like squirrelfish, yeah, they'll eat some, but really the really evil predator of these things is live coral. So if you put a gnathed anywhere near the polyp of a live coral, it sucks it in and eats it, because nathids are effectively zooplankton. When they're looking for hosts swimming around, they're effectively zooplankton. And so corals can nab them. And so we have video of coral eating these things. You can see the nathid gradually be sucked in, just like the Wicked Witch of the, which one is that? East or west? I always get confused. Yeah, she melts. So, so coral is a huge predator on these things. So you're probably thinking, coral predator, corals used to be abundant, but they're not so much anymore. What's going to happen with the native populations? So if you do experiments where you set fish, these are the fish inside this tube, and the nathids are actually attracted to the host by smell. They can smell the fish in there. You put a tube on live hard coral, you get very little. You put a tube on a dead coral head but still intact, you get more. And if you put a tube on rubble where the coral is totally degraded, you get a lot. So the more degraded the coral, the more nathids you get. And so wherever we've looked, and no matter what trapping technique we've used, we found the same result. That is high coral cover, low nathid abundance. Low coral cover, high nathid abundance. It's very consistent, very strong pattern wherever we go. All right, I'm going to switch gears now and talk about the role of parasites in influencing whether or not an introduced species can become an invader. So you may know that not all introduced species become successful invaders, right? They could be introduced somewhere, but situation's not right, so they don't proliferate. But others are introduced, and they go crazy, right? Their populations boom. And one of the most infamous invasive species in the Caribbean is lionfish, right? Everyone knows about that. So this is a collecting trip in the Bahamas. On a single dive in the Bahamas, this is the foot of my PhD advisor, Mark Hickson. Long after he was my advisor, we were collaborating on a project. And so this is one of 3 coolers we have on this boat. A single dive with 2 people, that's how many lionfish we caught. When I went to the Philippines and Guam, where these things are native, it took me, I spent a month in Guam and I got no more than 16, a whole month, working every day to find them. In the Philippines, I managed to get 30, but it took me over a month to get my 30, which is my permit lab. So they're far less common where they are native than where they've invaded. That's the trademark of an invasive species. So what is it that allows them to become so successful? Well, there are a variety of factors, but parasites can be one of them. This is a complex diagram showing all the different ways that parasites can influence whether or not interspecies is invasive. And one of the ways is that an invasive species may leave behind the parasites that infected at home, and when it gets to its new place, those parasites don't recognize it because they haven't evolved with it. So it's like, who are you? Get out of here. I don't want to invade you. So they leave them alone, and yet they still impact the local hosts. And so if you think of parasitism as a tax, that means that the introduced species can be essentially tax-free or much tax-reduced compared to the local species, and therefore have an advantage in terms of growth and reproduction. So that's what we looked at. So these graphs show are comparing lionfish with other fish that are in a similar trophic guild. That is, they eat similar things. Other kinds of predatory fishes like small groupers, snappers, grunts, things like that. Things that have similar. Most recent projects is looking at the biodiversity of 2 groups of parasites, the nathid isopods, which we talked about a lot today, and also the apicomplexans, which at least some of these nathids transmit. So I'll tell you that we're sampling throughout the Caribbean and Philippines, South Africa, Australia. Wherever we go, even within the Caribbean, different islands, we find different species of these things that have no name. They're not described. Nobody knows what they're called. So they're brand new to science. So that's kind of mind-boggling. You wouldn't think about going to different islands and seeing new species of fish, right? It's like the same ones throughout the Caribbean. But every island we go to, we find different nathids. And then the blood parasites are even more mind-boggling, because when you start looking at their taxonomic affiliation, you quickly realize there's no place for them. Like they don't even fit well into any of the known families of apicomplexans. And so they're probably an entirely new family, which is mind-boggling. It's, finding a new species, it's not that common. Finding a new genus is pretty rare, but a new family is just like mind-boggling. And so we're really looking at things that are virtually unknown. And so we're characterizing the biodiversity of these organisms. And so our collecting efforts here this week are targeted toward these things, the aphids, which we catch by a variety of means. One of them are light traps. We can use immersion traps. We can put fish in cages and put them on the reef and pull them up at dawn. We can take chunks of the rubble and put it in fresh water and flushes them out. So we have a variety of ways we can work depending on the location and what's feasible. All right, so one of the things we get to do is when we find and discover new species, we get to name them. That's the beauty of working on things that are diverse and that nobody else is working on. So we've got about 50 undescribed species in our catalog, and it takes about a year to get through to work up a complete description. By the time you do all the drawings, the molecular characterization, everything, it takes a long time. So we got a huge backlog of that. And it's getting worse because we're collecting more samples. But we love the arts. We appreciate what the arts can do for science. We appreciate the ability of artists to connect with the public. And we're music junkies. We love Bob Marley and the Wailers and other musicians. And so the first one that we described from the Caribbean was Nathia marleyi, which we named after Bob Marley. And the most recent one, which the paper just came out in June of this year. Oh, I'm sorry, let me backtrack. So through the naming of Nathia marleyi, I met the bass player for Steel Pulse in a hotel lobby in St. Martin about 10 years ago. He had heard a bit that some guy had named a species after Bob Marley, and he wanted to meet him, and somebody else there told him that guy was going to be showing up there tonight. So we met in the lobby and we had a long conversation. He filmed it. That conversation is actually online. You can find it online, the whole conversation. And so we became friends. And so Amlak Tafari is his name. Plays bass for Steel Pulse. And backtrack. We've inspired other artists. So this one on the left is a shirt that's made by an artist on St. John named Sloop Jones. It's our Nathia marleyi shirt. And then we have people who've painted Nathia marleyi. So a lot of interest among artists. The most recent one that we named in June was after Jimmy Buffett. And before he passed, he knew about this. He heard about it, and his comment was, it has a nice ring to it. So, we feel fortunate that he knew about it. Very sad, though, that he didn't stick around to really appreciate it and enjoy it. So that's our most recent Nathan paper. All right, now I'm going to switch gears and go back to our roots. How we got into parasites was by watching cleaner fish clean fish at dawn. Now we're going back to cleaner fish and looking at what else can cleaner fish do. There's a lot of interest and research now going into microbiomes. Microbiomes are the suite of microorganisms that cover every surface, living and non. If you look at your skin and you ask, whose cells are these? Most of the cells on your skin are not your own skin cells. They're microorganisms. And you've got microorganisms in your gut, all over. So they play a huge role in all kinds of processes, including behavior. And so, and because cleaner fish physically contact hundreds of fish a day, different species, and they're also in contact with the corals. They have the potential to spread lots of microbes. And so we're looking at that. And so here's a cartoon of how cleaner fish might influence the diversity of microbes on coral reefs by basically connecting everything else in the picture. Just like going to a party and the host is contacting everybody, all the guests. That host is putting all them in contact with one another by that contact. So that's what you see on the right here. That's what's happening. All right, some quick results here. We've worked in 2 study sites. We did experiments where we removed cleaner fish from some areas and not others. And then we looked at the microbiome of damselfish that live around those cleaning stations. And in Puerto Rico, we didn't see any difference, but in St. Croix, we found a big difference in the microbial diversity of damselfish that had cleaner fish present compared with those whose cleaner fish we took away. And don't try to read these graphs. I'll explain them to you. We did experiments in the lab using ocean surgeonfish. And some of the ocean surgeonfish we soaked in antibiotics for like 5 days to strip their microbiome. Other ones we didn't do that. And then but in both groups, some of them were then exposed to cleaner fish and others not. The ones that were exposed to cleaner fish, their microbial diversity increased dramatically over the course of a week, whereas the ones who didn't have cleaner, access to cleaner did not. So the cleaner fish seemed to have a big impact on the microbial diversity of these surgeonfish. If you look at corals, effects on corals around this is just for St. Croix, around the coral heads where we had cleaner fish present versus absent, the number of visits to those cleaning stations was positively correlated with the microbial diversity, especially for diseased corals. So the impact was much greater for diseased corals than for healthy corals. And don't try to read this either, this graph. What I'm showing you here is that also there were more types of bacteria that were enriched where there were cleaner fish present than where there weren't. There are certain species of bacteria that were really characteristic of a cleaning station that were rare or absent on non-cleaning stations, suggesting that cleaner fish do have an impact on microbial diversity at cleaning stations. All right, so I started my career working on sharks, and then I got into smaller reef fish, mostly damselfish, and then I got into the parasites that feed on those damselfish, and then I got into the microbes on those fish, and we're also working on viruses. And so we're getting smaller and smaller and smaller. And the more we get smaller, the more we realize just how important those tiny things are. And so if I can borrow a quote from a Grateful Dead tune, again for the deadheads in the audience, once in a while you get shown the light in the strangest places if you look at it right. So when you start looking for these tiny things, you find them and you realize, wow, like they're everywhere and they're super important. All right, so I'd like to thank our sponsors, including See and Learn, for bringing us back this year. And the number of people involved in this work is just way too many to list. There have been hundreds over the years. Again, Gina Hendrick has been heavily involved in this work, and she's here today, but to list all of them would be virtually impossible unless I had 20 slides. So we've really benefited from all the help we've gotten around the world. We work throughout the Caribbean, we work in the Philippines, Australia, South Africa, Hawaii, anywhere there's reefs and parasites, we work, and we've built teams that are active in all these places. So we thank all of them, and we thank you for listening. Questions? Yeah.
Who provides your funding?
Most of it comes from National Science Foundation. Who provides our funding? That's the question. And the answer is most of our funding comes from the U.S. National Science Foundation. So our earlier work was funded by Biological Oceanography Division, and more recently we've been funded by Biodiversity and Systematics Program.
Any other countries providing?
Yeah, they provide support on their end. So when we go there, they provide some support from them, and we provide our support with NSF. Typically, theirs isn't as much as ours, but they definitely contribute. Yeah.
If I understood you correctly, you said there's more parasites on reefs that are degraded.
Well, at least certain kinds. Yeah.
And you probably want to repeat the question, but
Yeah.
do you think that's because the parasites already did damage, or is there any hypothesis of why?
Okay. So the question was that I mentioned that parasites were more abundant on degraded reefs, and I'll qualify that by saying certain kinds of parasites are. The general pattern that you see in nature is that a healthy ecosystem has a very high diversity of parasites, but no one gets super abundant. So you don't have huge numbers of anything, but you've got lots of things, or lots of things you have a small number of. And when things get disrupted, what you end up with fewer species of parasites, but those that are there go nuts for various reasons. So with respect to the gnathid isopods, and this also applies to other parasites that have free-living stages where they have to find a host by swimming, and they spend a lot of time in the bottom. That includes things like monogeneans and even some digenean trematodes. Anything that's got a stage where it has to move to get to a host. High coral cover, the coral eats all those things. It'll eat monogenean larvae, it'll eat digenean larvae, gnathids, whatever. Coral is like, they feed on everything. So high coral eats those stages, which means you have fewer of them that can infect the host. Once you lose the coral, as long as you still have fish hosts, you've got nothing on the bottom that's going to do them any harm. Like all that bottom is prime real estate. It's rubble. They love it. There's nothing there to eat them. And so you get these spikes. And again, the pattern we see is dramatic. But what's really interesting is that it only takes about a fairly small amount of coral cover to have an effect. Even something like less than 25% coral cover will have a significant impact on the abundance of these parasites. And once you get below that, things can get really, really high. So if we want to go catch gnathids or monogeneans, for example, we know if we find areas where there's lots of fish and very little coral, we'll hit it every time. So, yeah. That answer your question?
Yes, sir.
Yeah.
So in addition to the degradation of coral reefs, seagrass bed loss, how does that affect the health of fish that are affected by
That's a really good question, and we really haven't looked at that much. So, the question was seagrass beds. So we talked about how degradation of reefs impacts the abundance of the parasites. What about degradation of seagrass beds? And we really haven't looked at that. That's a really good question, actually. Yeah, we've did some sampling out there with our light traps and with fish cages, and we do catch them out there. We've not really quantified how what we collect differs among seagrass beds that are in different conditions. So thanks for the idea. We'll do that. Yeah.
Do we know of anything, or is there anything to be learned about competition between the parasites?
That's also a really good question. So, the question is competition among parasites. So is there anything to be learned about competition among parasites? And so internally, that's definitely the case. So internally, parasites have their spot where they like to live, and they don't get along well with other ones who want to live in that same space, whether it's in the gut or inside the body cavity, whatever. There's some competition there. For ectoparasites, as long as there's surface area where one can attach, it doesn't matter, because they don't. Once they attach, they're stuck there and they can't move to fend each other off. So internal parasites, that is the case, but probably not for external parasites. Anything else? Yeah.
We dive a lot. So as divers, can we be affected by these parasites?
The question is, as divers, can we be affected by these parasites? And the answer is no. So we swim with them all the time. We handle them, and they don't do anything for us. Yeah. So no worries there. Yeah.
You mentioned that you had that the infected fish, it does not go to the grassy area. They stay on the reef. What's your perspective when you have they're staying on the reef? If they have their parasites
Yeah. So it's a double whammy. So they have that big one on them. That from other studies, it's been shown that creates a significant amount of drag on the fish as it swims. So it's no shock that they don't make the trip. And yet they're staying put where all the gnathids are. So it's likely that they're getting hit pretty hard, And so fortunately for them, those antelope only last for about a year, and then they die and fall off. But that 1 year could be pretty rough. So what we haven't done is actually quantify that. not only the extent to which they get hit more, but also how that impacts their longevity. We haven't tracked them long enough to really look at that.
But aren't they getting some advantage of being still on the reef where there's anything that drops off? Paul, could you repeat the question?
Yeah, so the question was, wouldn't there also be a benefit because the parasites that fall off would be eaten? And so that could be true if you have a high coral and lots of coral. Yeah, but when you don't have much coral, you don't get that. Yeah. And they tend to live right on the edge of the reef, not deep in the coral, so on the edge on the sand reef interface. So they probably don't benefit much from that. Good. All right. Thank you, Tech Man. Are you mean like movement of Oh, absolutely. Lots of things you can do. There aren't, I can't think of any parasites that go from, well, they might be something like crabs, like the land crabs that go from the ocean. There's probably something that goes, in them that goes from the ocean to land cancer. But in general, because the climates are so different, not so much. But there's a lot of things that you can learn into. So a lot of what we